I'm delighted to introduce Jan Kulagowski, who is with us to talk about an example of the Virtual Biotech's involvement in drug discovery and preclinical stages of finding new treatments that we need. So with that, over to you, Jan.
Thanks very much, Chris. And I'd like to thank both you and the Peninsula Parkinson's Research Interest Group for giving me the opportunity to highlight one of our virtual biotech projects I've been heavily involved in. So I'm aware that perhaps some of you don't know me very well, so perhaps I can just start before we get stuck into the presentation with a bit of background on myself. I obtained a PhD in organic chemistry from Imperial College and then undertook postdoctoral studies at Stanford University in the US. On my return to the UK, I joined US pharma company Merck Sharp & Dohme at their newly opened Neuroscience Research Center, where I spent many years working on, amongst others, stroke, pain, depression, Alzheimer's, but unfortunately never Parkinson's. We did manage to get a selective dopamine receptor blocker into the clinic, but this was for schizophrenia and not Parkinson's, although I was interested to see more recently that that particular drug is being currently investigated for its potential in levodopa-induced dyskinesia. So after Merck Sharp & Dohme, I moved to a contract research service provider where I helped manage projects in a variety of different areas before joining Parkinson's UK in 2015. And I have been involved in the development of the virtual biotech from the very outset, which has been a particularly gratifying experience. And as a drug discovery manager at Parkinson's UK, I divide my time between searching for and assessing potential new projects that we can bring into the virtual biotech, as well as helping manage some of the existing ones that we already have in our portfolio. So as Chris mentioned, the project I'll be describing to you today is one that we have been working in partnership with a biotech company called NRG Therapeutics. And the project is aimed at, let me actually share my screen. I've forgotten I'm not showing you my slides. So as I said, project with NRG Therapeutics, and this project is aimed at developing a drug which acts on mitochondria inside brain cells. And mitochondria are found in every one of our cells in our bodies, and they produce the energy that the cells require to function properly. And we know that in brain cells affected by affected by Parkinson's, I beg your pardon, the mitochondria don't work properly, and they don't produce sufficient energy for the cells to be maintained. And also the malfunctioning mitochondria can cause problems themselves. And as such, this area is a very active and important field of research leading to potential treatments for Parkinson's. There are many groups working around the world in this area. So what was it that drew us to work with NRG in particular on this project? We were introduced to NRG by a mutual contact and were very impressed by the team and their ideas. At the time, the company comprised only 3 employees, and they were searching for funding in order to support a novel approach at maintaining mitochondrial health. However, they were finding that their ideas were too applied for academic grant style funding, but the project wasn't sufficiently developed to attract funding from a larger investor. I should point out that the company's name is very apt for an organization interested in mitochondria, in that it spells out energy, and the initials are, and the letters are also the initials of the 3 founders, namely Neil, Richard, and Grant. In addition to producing energy, mitochondria also control levels of calcium within the cell by absorbing any excess which could become toxic. And when cells die in Parkinson's, the remaining dopamine producing cells have to work extra hard, and this puts the mitochondria under stress, and an overload of calcium and an inability to produce sufficient energy to maintain the cell leads to opening of a pore on the mitochondria called the mitochondrial permeability transition pore, or MPTP for short. And opening this pore then starts a process of cell death and loss of further neurons. So we funded, or we invested £1 million in the project, in NRG, to help them set the project up in 2019. And at the time, there were already some molecules that were able to block the MPTP. I should have said, actually, to the previous slide, that NRG's approach is to find a drug that stops the MPTP from opening. And this is a distinct approach to that of other researchers in the field. So there were already some molecules known that could do this, that were in the literature, but these were very far from a potential drug. And in particular, they weren't able to get into the brain, which is, of course, a specific requirement for any drug destined for Parkinson's. And so the team started looking at these along with a research contract service provider. They made new examples which had better activity, perhaps less potential for toxicity, and importantly, were able to get into the brain. Alongside that work, they also undertook a highly automated testing of 200,000 molecules to find completely new molecules. And this provided a number of hits, but these were further back in the process, which was very exciting. But these are very early stage projects, very early stage starting points. And so the team put plans in place to help craft these molecules into something that better resembles a potential drug. We were very encouraged by the process, by the progress made on the project, and invested further in the company to allow them to improve molecules from both of these starting points. And this process involved a cyclic, iterative process where new molecules were made, they were tested, the results analyzed, and then used to design the next round of molecules, and so forth. And in this process, a number of tests were conducted to see how good the molecules were, particularly how effectively they prevent the MPTP from opening, whether they have potential for undesired side effects, whether they would be able to survive being in the body for any length of time, and of course, whether they get into the brain. And another very crucial aspect of this work was to ensure that the molecules were unique, that they were novel. And this is a very important consideration when it comes to trying to attract further funding. And so the team set about patenting their new discoveries. While the MPTP has been known about for considerable length of time, its actual identity remains a mystery. And it was felt that knowing exactly what their molecules interact with in the pore would be a major boost for the project. So the team started to look at what this could be. And if this work is successful, then it would represent a major scientific breakthrough in mitochondrial research. Given the excellent progress made on the project, along with our initial funding for the project, the team were able to secure further investment more recently. So this was in the form of 2 Michael J. Fox Foundation grants. So Michael J. Fox, as you know, is a US Parkinson's charity, amounting to $1 million US. They also received a very generous grant from a government-supported UK funding agency. And late last year, we're also able to announce the raising of a Series A venture capital investment fund of 16 million pounds. So this shows the value of the virtual biotech in that by injecting a relatively modest amount of charity funding and deploying it at exactly the right time would allow a project to progress to the point where it becomes attractive to larger investors. And of course, also this vindicated the virtual biotech's belief in NRG and their project. With this very substantial injection of funding, the company is expanded. The original team have brought in further expert researchers and scientists to help drive the project forward, as well as a management team that will help keep it on track. And it's hoped that with continued success, this funding will allow the project to get to the point where we can think about entering clinical trials, and continued success will hopefully lead to further investment, which would be required for these very long and complex clinical studies. So it's been a pleasure to be able to tell you about one of our particular projects in the virtual biotech, and this, as you've seen, is a earlier stage project than the one you saw from my colleague Shona earlier in the year. And I hope also that I've given you some insight into what happens at an earlier stage in a drug discovery campaign. We've been very encouraged by the progress made on this project, and it's certainly one of the shining lights within our virtual biotech initiative. However, we know that not every project will be as successful, and in fact, even with NRG, there are no guarantees. As such, we're always looking for new projects to bring in, and because we understand as scientists that the path to a successful therapy for Parkinson's is a very long and unpredictable course. And that is the power of the virtual biotech, in that we can bring in multiple projects and support them simultaneously. Those that continue to be successful, we will continue supporting. Those that perhaps don't make the progress we hope for, we can let go and bring in new ideas. And it's only by exploring multiple avenues and seeing what works and what doesn't work equally will enable us to ultimately find the answers to Parkinson's that we so desperately need. And with that, I will thank you for your attention, and will be happy to take any questions.
Thank you very much indeed. I must say that you've clearly put your shoulder to the wheel of explaining this to the man in the street. I found that most illuminating. Thank you very much indeed.
This may be a very, very basic question, and therefore I apologize for that. But I'm hearing and sort of understanding a whole sequence of very complex stages towards results. But in my naiveness, I'm saying, why in Parkinson's, why are there so many complex stages compared to, and I asked a similar question, the more simple task of COVID? Is Parkinson's so much more complex a condition compared to most of the conditions that people get in general medical environment? And so this is quite an exception to the normal route of medical care.
It's a very good question, and I would say that yes, Parkinson's is far more complex. We don't really understand as much as we would hope to by this stage as to what causes it. As you know, most cases of Parkinson's are idiopathic. We don't really know what the cause is. And so, as with most conditions that affect the brain, it's particularly difficult. And that's shown by the fact that there's such a plethora of treatments for the condition. You mentioned COVID, and so I've been in the business of drug discovery for all my career, and it's longer than I would hope to remember. But COVID wasn't the work done that we were all so impressed by a few years ago in bringing forward new treatments for a new disease was exceptional. And from my experience, in general, bringing a new medicine to the market would require something of the order of 12 years of research and up to $4 billion US to get it from the point of inception to the market. So it's always a long process. What we saw with COVID was an exception, and we were able to do that because there was already quite a lot of background research done on this new type of vaccine, and there was an enormous effort by pharma companies, by governments, all working together to try and get something as quickly as possible. But that is exceptional. But Parkinson's is a particularly complex condition, and it's probably not just one thing. It's probably, as you yourselves know, different symptoms. Everyone says their Parkinson's is their own. It's theirs. It's different to other people's, and that makes it all the more complex. But thank you for your question. Really good question.
Thank you.
I have not so much a question, I suppose, as much as an observation. It is very clear to me, at least I think it's clear, that the only thing that makes the world go round, makes this work at the end of the day is money. Mm-hmm. If there is no money, there is no drug, there is no progress. No matter what we might think of that from a moral point of view, that is a reality. And it's how difficult was it for a charity to accept that it had to create wealth in order for the process to happen?
Again, a very good question. Yes, there are people who do think that working in partnership with commercial concerns is not what a charity should be doing. But previously, and we still do this, as part of our research activities, we fund academics to do the very basic research. But unfortunately, most academics aren't that interested in developing a drug because it's a very applied, quite tedious process, if I can say that. And it's really down to the companies to do so. So yeah, it was I think a tough decision, but I think the right one. But what I should point out, yes, we are providing commercial enterprises with funding, but in all of our contracts, we have an agreement that once they become solvent, once they make a profit, we would gain some return on our investment. Right. So it's not like it's a one-way process. And that's not the most important thing. The most important thing for us is helping people with Parkinson's in whichever way. And in the research team, we do it by, as you've seen, trying to develop new medicines amongst other aspects. And so, yeah, it's a necessity really to work with these people.
Chris, can I ask a question?
Yes, Sue and then Nigel.
Excuse me. One observation is how important it has been to choose founders of a company with the correct initials. I don't think I will always any time forget NRG. And also it's good to see people ploughing money into original research rather than just repurposing. Repurposing drugs is attractive because it shortens the length of time to develop it. But once you've exhausted all those drugs, what's coming on? And so far, repurposed drugs haven't been the bright light at the end of the tunnel. They've been just a little weak light bulb halfway along. So well done to Virtual Biotech there.
Thank you very much. Again, you raise a really, really good point. When people hear about the potential for repurposing a drug, they get very excited because they think all of that very tedious stuff that I just tried to give you a flavor of, and that's only a very small part of what goes on. You know, you don't have to do because the drug's already on the market. And there are cases where a known drug has been repurposed for a different use, but it's not made bespoke for that, particularly for something like Parkinson's, where, as I explained, the drug has to get into the brain in order to work, and that's a really difficult thing to do. So most drugs don't get into the brain, particularly if they haven't obviously been developed for a condition that affects the brain. However, and so the other aspect of research into repurposing is that by the time a potential new use of an old drug is found, the drug is off patent. So it doesn't, if you like, belong to anyone. And so it's a free-for-all, anyone can work on it. And that doesn't bode well for getting investment because there's no return, because it doesn't really belong to anybody. And so it's very difficult to attract investment for a repurposing effort. However, as a charity, because as I've just said, our main purpose is not to make a profit, but to help people with Parkinson's, we are able, if we feel there's a really good possibility of taking on a repurposing effort. And in fact, within our clinical portfolio that Shona looks after, there are 2 such projects. And we'll see whether they're successful, but we felt there was sufficient potential to fund clinical trials for these 2 particular drugs.
Thank you.
Thank you. Nigel.
Hi. Is there any commonality between brain disease, i.e. sort of MND, MS and Parkinson's, for example, with regards to the initial pathways? And does Parkinson's UK sort of investigate with other organisations regarding commonality?
We do. That's, again, really, really good questions. I'm really delighted with these questions. We do work with other organizations, but obviously with our very limited resources, we can't start looking into Alzheimer's or other conditions. But what's really interesting in all of these neurodegenerative conditions, of which Alzheimer's, Parkinson's, and others are part of, there seems to be an errant protein or a well-known protein which misbehaves, that is thought to perhaps contribute to the progression of the condition. And in Parkinson's, it's a protein called alpha-synuclein that you may have heard of, and in Alzheimer's, it's something called beta amyloid. And that's a really interesting area because there's a huge amount of research ongoing into whether alpha-synuclein causes Parkinson's, or is it just something that happens after the condition kicks in. And looking at research that's been undertaken in Alzheimer's, which has been going on for much longer than in Parkinson's, they've been looking at the errant protein in that case, the amyloid protein I mentioned, and after a very long period of research, over 30 years and a huge amount of money spent, it's still not clear what the situation is, although I think the first antibodies to that protein have been approved recently, so we'll see whether they are actually effective in treating the disease. What people really look for in terms of trying to find a mechanism of action that they can latch onto and find drugs for is a genetic link. So if there is something within our genes that make us more prone to getting a particular condition, then that's a really good starting point, because we can then start looking at that. And with Parkinson's, that's not the case. There is a very small genetic component, probably less than 10%, where you can trace the condition to a genetic mutation. I think the situation is more prevalent in Alzheimer's, which is why there's been so much interest in amyloid protein, because there is that, or it's felt that there is that genetic link.
Clearer genetic link.
Yeah, so people felt it was a really good process to modulate through some pharmacological interaction. But as we've said so far, it's not proven to be quite that easy.
Okay, I have one more question, which I think wraps around to Harvey's introduction, which prompted you to confirm the complexity of the disease in the first place. So I'm looking at the complexity of resolving a situation with a particular person like me, one of the 9 of 10 idiopathics. The implication of this is it's not going to take one thing to fix me. It's going to take 2 or 3. And by the way, they'll all be too late, because this will be I shall be history by the time the farmer has produced the goods. But never mind, it's still going to take 2 or 3 or 4 things to fix people like me. How do you test that and to the satisfaction of the regulatory authorities?
I'm probably not the best person to answer that question. That's right at the other end of the drug discovery continuum to where I tend to operate.
You can hand it on to Claire then.
It's tough. If it's going to be a cocktail of drugs that are required for everybody, then that's going to complicate the matters, obviously. But I guess we'll have to take it stepwise, and so find a treatment for a particular aspect of people's Parkinson's and then work from there. But the regulatory authorities are there to help. Sometimes they seem to get in the way and slow things down a little bit, but they're there to help. And so if there is a clear benefit in either a cocktail of drugs or one particular medicine which works for an aspect of Parkinson's, and if they see there's a clear benefit, then I think it will be possible to get it through. But clearly in this country there is an element of whether it's going to be worth the money, because some new treatments are very, very, very expensive.
And on that thought,
I was just going to add, there is some sort of precedent for that kind of poly pills or like polypharmacy kind of approach, I think, in areas like cancer. Cancer is always further ahead than us, right? Mm-hmm. So, and it is something that I think is a rumbling story that's not quite taken off yet for Parkinson's, but I think you're right, we'll get there, and it maybe will tie in with our ability to look in more detail at the biology of each person. So, for some people, we might be able in the future to look somehow at blood test and say, okay, so this person has an alpha-synuclein heavy version of Parkinson's. We can see the mitochondria are struggling. We'll give them these 2 drugs that fix those problems. Another person, the biological drivers of the condition might be a bit different, and then maybe they need something slightly different. But that's like, that's the dream.
Well, absolutely, that's a good point, and that speaks to personalized medicine, where everybody is evaluated for what it is that's going wrong with them, and then the treatments are sort of applied bespoke to them. And as Claire has said, it's becoming quite commonplace in cancer, which is better understood but still requires an awful lot of research, and there's more of a genetic link there as well, so maybe it's slightly easier to do that. But that would be the dream, but it's a long way away. I'm not sure any of us will actually see it in our lifetimes, but let's hope that it comes sooner rather than later.
Okay, and with that thought, lifetimes, I'd like to close the proceedings. And thank you all for your input and your care and appreciation. It's much appreciated. Thank you all.